voice, or you can write it in the little chat window as well if you want. Both those options are perfectly fine, and we will answer both of those. And we will be taking questions when we're done. How this is going to work is there are going to be two parts. First, we're going to give our little talk about the topic we're going to talk about, which is, yeah, you'll see, it's cool. And then we'll go on and we'll actually talk about the program itself, and we'll answer a question at the end of each part. So first, we will do our little presentation, answer some questions you might have about that. Then we'll talk about the program and answer some more questions. And we'll see how it goes from there. Okay, so let's introduce. I am Ryan, and this is Jessica. And I guess if I point down, it's you, but I don't know if that works for everybody else.
I'll just wave.
We are the coordinators for the Bioinformatics program, and we will start talking now. Then Jessica will start off.
Oh, okay. Hi, everyone. Nice to meet all of you, and welcome to our session. Today, we're going to be talking about a news article that was published in the New York Times. It's a short and sweet one-figure article that talks about virus mutations around COVID-19 specifically, and how we can use the information about viral mutations to examine how viruses spread around the world. And Ryan has done us a great favor, and he's actually posted the link to the article in the chat. So if you want to follow along with the actual article itself, feel free to click on that link. But we're going to go through it pretty briefly, but certainly with lots of nerdy intentions here, I think, right, Ryan?
I hope so.
I'm going to deal with the biology side of things, and Ryan's going to talk more about the computer science side of things. So the title of the article is Virus Mutations Reveal How COVID-19 Really Spread. So the first thing I want to do is just define for us what exactly is a mutation. Some of you probably already know what it is, but just so we're on the same page, a mutation is a change of the structure of a gene that results in a variant form that could actually be transmitted from one generation of an organism or a virus to the next generation of an organism and subsequent generations thereafter. You can have a mutation that's just a very small single base pair nucleotide kind of change, or it can be chromosomal. We won't go into that much detail here today, but there's lots of different examples of how what mutations look like. We're going to focus on the single small nucleotide changes that are considered to be mutations for talking about COVID-19 today. And lots of things can trigger mutations to occur: chemicals, exposure to physical agents like UV and radiation, and that sort of thing. The thing about viruses is that they tend to mutate and mutate often. We'll talk about rate in a minute, but they tend to mutate very quickly and often. And this is because viruses have this really neat way of replicating. They need to get into a host cell, and what they do is they hijack the host's machinery to be able to make more copies of itself. And a virus is really mostly DNA or RNA or some kind of nucleic acid. Sometimes it's covered in a protein packaging, sometimes it's a lipid packaging, but the most important part of the construction of that virus is its nucleic acid. In the case of COVID-19, it's an RNA virus. So that virus can't replicate on its own. It goes into a cell, it hijacks the cell's machinery, and it makes more copies of its nucleic acids, in this case RNA. But because it does so rapidly, the process is error prone. So viruses tend to mutate much more than human cells, for instance, and even bacterial cells. Most of the mutations that happen in a virus are redundant because it happens in the sequence somewhere along the way that doesn't really code for anything, and therefore that change doesn't make a difference in the functionality of that viral particle. That's called a redundant mutation. Sometimes the virus can mutate in the part of a gene that is important, in which case it often breaks the virus, so to speak. What I mean by that is that the virus can no longer replicate, and that's the end of the virus. So in both of those situations where there's a redundant mutation or a mutation that causes the virus to just sort of fizzle out, really no big deal. But in the case where a mutation occurs in an important gene and it actually gives the virus a new or novel function, can create a new strain of the virus that has a different physiology. So that virus can go on to infect a broader host range, or it has better infection processes, or so on and so forth. Right now, when we're looking at COVID, COVID has mutated. And it's mutated in such a way that most of the mutations that we're seeing in the strain of COVID-19 are the redundant form, so that there's changes, but they don't really make a physical or phenotypic difference, so to speak. So one of the things I said is that viruses tend to mutate and mutate often. I wanted to sort of show what that means. So this diagram here that I'm showing everyone on the screen looks at the genome size across the x-axis and compares it to the mutation rate as a function of mutations per site per year. So if we look at this image, eukaryotes, the cells that make up mammals such as us, they tend to mutate or have large genomes, and the mutation rate is quite low because eukaryotes have these editing processes that if a mistake happens, we can repair those mistakes. Bacteria, smaller genomes, higher rate of mutations because they lack some of those editing processes as well. And then over here, we've got the RNA viruses, of which COVID is an example. So the RNA viruses are very error prone. Viruses don't have the machinery to repair mutations, so they tend to have a very small genome with a high rate of error or mutation rate. So when we're thinking about COVID-19, just to put it into perspective, COVID-19 is hypothesized to have an accumulation of 24 substitutions per year. It's got a 30,000 base pair genome, so it's a small genome, and it's got a pretty high substitution rate per year. And if we compare it to something like influenza, influenza is 2 mutations per year, or HIV is 4 mutations per year. Humans, per base pair per year mutation rate is 0.5 times 10 to the negative 9. So relatively low, very, very low rate of mutations. The neat thing about these mutations is that we can use the mutations and look at the changes in a viral nucleic acid, RNA in the case of COVID-19, and we can examine how those mutations travel through space and through time. And what we do is we use a map that's called a phylogenetic tree, or a tool if you'd like, to be able to map that process. I'm showing you a very generic version of a phylogenetic tree here, and you may be familiar with phylogenetics. Typically we see phylogenetic trees when we're looking at evolutionary relationships between among organisms or between species. So for instance, if we were looking at the human phylogenetic tree over here, we'd have primates and then Australopithecus, and then up here somewhere we'd have the hominid species, and you'd see how the family kind of branches out and how some end and others don't. So really we would be looking at different species. In the case of a virus, we're not looking at different species per se, but what we're mapping here are the mutations. So what this diagram is showing is we started out over here with our novel COVID-19 coronavirus. And in about a couple of weeks, what happened is it developed 2 different mutations at 2 different sites. And so now we've got COVID-19 still, but it has a mutation here and a mutation here. And then this guy mutated even more. So it's got a new mutation here and a mutation here. And then it started to create these different strains within the same family. I'm going to come back to that word strain in a second. What the phylogenetic tree shows us is it maps over time how those mutations accumulate, and we can use this information then by, for instance, if I've got a sample of coronavirus, I can compare it to this phylogenetic map and I can see, okay, my sample maps to the strain that is called E in this diagram. And I can kind of figure out, okay, when did this particular strain arise and where am I seeing it proliferate in the world? So that's a simplified version of what a phylogenetic tree looks like. And I use this word strain. So in the case of strain, right now, so as of from December to May, there's been 5,000 or so COVID-19 genomes that have been sequenced. and we're still considering it to be one strain. Although it's developed all of these different mutations, it hasn't changed functionally to be able to call it different strains. So I just wanted to clarify that terminology right from the get-go. Okay, are we ready to look at the phylogenetic tree for COVID-19? It's scary, I'm warning you guys, but here it is. This is the one directly from the article that shows us that we started over here with COVID-19 somewhere in December in China, and every dot that you see in this picture represents a mutation that has occurred. And you can see that there's color coordination happening here, where a number of mutations took place very early on, and they're sort of classified under pink. The size of the dot represents how many genomes fall in that category, so the size matters as well. But you can see that there's been a number of pink mutations. The majority of those pink mutations occurred in the region of China. So then when we follow along from December and move into January and then into February, COVID-19 started cropping up around other places in the world. And when those genomes were sequenced, more mutations started to appear or be illuminated through sequencing studies. So if I just focus down here on these yellow guys, most of those mutations occurred when the virus traveled into Italy, and then several branches came off of that from the perspective of where those mutations, the sequence mutations were found. And similarly, we can see blue up here in Europe, and then in the rest of US, we see these orange dots sort of proliferate throughout this phylogenetic tree. So what I said earlier is that we can use this information from phylogenetic trees to ask questions like what is the epidemiological spread of this virus around the world? So what I want to do is I actually want to navigate over to the site where we find this information. Here we go.
I posted a link in the chat if anybody wants to jump over to it as well.
We follow along. Yeah, it's called nextstrain.ca. So the one that's from the article only shows us a snapshot from December to March of, I think, March 25th or something like that. The latest data you can find from the original website, which is what we're looking at here. And you can see along the bottom that now we're looking at December 3rd all the way up to April 29th, the most recent data. So just a bit about this site. This is an open access online research portal. It's called NextStrain. And this group, which is a multi-center group from all over the world, so the institutions all over the world collaborating together, what they've been doing is they've been collecting and curating viral genomes ever since 2015. So before COVID even happened, they were collecting viral genomes to understand the epidemiology and see how we can use the information to manage some of these diseases. But from December to May of this year, they've collected 5,193 COVID-19 specific genomes that have been publicly shared, collected from scientists and medical doctors around the world. Now, if you think of that, that's an incredible feat. Between December 2019 to today, 5,000 COVID-19 genomes were sequenced and then uploaded to this collaborative shared site in the midst of a pandemic. So that's crazy. And what it says to me is that this endeavor to collecting data and doing bioinformatics is a very social, supported, open access type of endeavor, and people are working all around the world to make sure that the data is out there and that we can use this data to help create some management strategies to control and treat this virus using either vaccines to stop the spread or antiviral agents. And I think that's an amazing motivation. Okay, so what we're looking at is the phylogenetic tree. Once again, really messy, but kind of beautiful at the same time. What these folks did at NextStrain was to take this information using an algorithm and to map it around the world to look at how the disease actually spread. Okay, so here's the map, and the colors represent the different versions of COVID-19, not strains per se, but versions of COVID-19 that are based on those mutations. So if I click play, what it will show us is how these versions moved from its origin point. So there it is, starting off in China. Everybody can see that first COVID-19 with a very specific genome. And then we start to see it cropping up in Thailand. You can see that tiny little dot there in blue. That one in Thailand had a specific subset of base pairs, so a little bit of a change as it moved away from China into Thailand, into the Middle East there, into Egypt, and then into the States. We see the red dot indicating that now we've got sufficient change, or we've got some more mutations that have occurred, and we've got a different version of COVID-19 existing in those areas. Yellow in Europe, you can see it coming up in Italy, in Spain, in France. We can watch as this virus moved across the globe into different areas. So I think this is fantastic information. And as far as we know, we're still working with one strain of COVID-19, and we know that that one strain, how that one strain has moved across space and across time. So by comparing these viral genomes to each other, we can characterize the evolution of the virus and the movement around the world. One thing to keep in mind is that this type of information, although it's really useful to infer some data, it's not complete. So remember, I said that we're really relying on scientists and medical doctors around the world contributing their sequencing to this particular group, nextstrain.ca. It doesn't mean that everybody around the world is contributing at the same pace. So for example, the map shows very few sequences that are coming from the global south, right? Typically, we're not seeing as many contributions coming from, say, South America. And it's not that COVID-19 doesn't exist there, it's just that the data hasn't been contributed by those areas. So while there's lots of really good information and trends that we can see, certainly, we just need to take it with a grain of salt. Okay, before I pass it along to Ryan to take a look at the magic, the computer magic that actually goes on to create these maps, I also just wanted to point out that this is the data that's related to COVID-19, but as I mentioned before, NextStrain has been collecting and mapping viral data for quite some time. So I wanted to look at one other example for a comparison, and it's very purposeful, the one that I chose. You've probably heard a lot of people saying that COVID-19 is no worse than the seasonal flu. And while for many reasons that's not quite true, I wanted to show you some of the same data, or a data set anyway, that looks at flu viruses specifically. So hopefully this will load for me. Oh, blank screen, that's never a good sign. Give it a second. It's loading. Oh, maybe it's not. See if I can get it this way. All right, well, oh, there we go. So here what we're looking at is the phylogenetic tree for influenza. And what you'll notice here is that we've got different names for influenza now. So we started over here with 3C2 and it's moved into A, and you might know of influenza B and so on and so forth. So we now know that influenza actually does have different strains, and each of these different strains are functionally different. They have a phenotype that is distinct from the other strain. So influenza A is different from influenza B. So there's that. And then when we look down here at the map, I won't play you the real-time data. I'll let you kind of look through that yourself if you decide to play around on this website. But I did want to show you this diagram here, which shows us the information about influenza that's going from October 2013 to date. And so if you look at the bubbles that correspond with the incidence around the world, and you compare that to what we saw with COVID, this is longer term data, and the incidence of influenza is substantially different from COVID-19, which has only really been traveling around the world for a select 6 months or so. So comparatively, we're looking at a difference in terms of the rate of mutation. So like I said before, it's only about 2 per year, 2 per substitutions per year compared to 24, which is what we see with COVID-19. And as well, we see a difference in terms of how it's spread around the world and prevalence in different areas. So a little bit of background information to describe how some viruses can be very prolific. And in the case of novel coronavirus, COVID-19, there's been a fast, rapid spread, lots of mutations that have been taking place. Okay, so I'm going to pass this along to Ryan now, and he's going to talk code.
So I'm just going to try to steal the screen. Okay, let's do that. And now, mess it up somehow. Hello? Can you hear me? Okay. And you can see the screen right? Awesome. Maybe there's a green border, but that didn't show up this time. But that's fine. Okay. So as it was talked about, this is a collaborative effort. All this stuff is openly sourced, which means you can actually go to the GitHub that has all the information that you might be interested in. So if you just click on the little link over here, it will take you to this, which has a lot of the code that went into this. Okay, so you can go through, dive through, look at some code, and I'll talk about some of that today. I'm not going too deep into this because there's a lot here. I'll talk a little bit about it. But before I get into anything really too interesting, let's look at the actual data. Okay, so Jessica pointed out how we were sequencing genomes. Hey, what does that look like? This would be an example of one. So starting from down here and going all the way up, that is one sample of it. It's a lot, it's big, it's scary. This is why bioinformatics comes into this. There's no way you can imagine that any human could look at this and make sense of it whatsoever. It's a block of text. Okay, so then we're going to feed this in, and this is the data we're actually going to use. And that's how we're actually going to build up these trees, and this is what we're talking about when we sequence the data. And to me, this is how I see genetics. It's a series of characters. Okay, so let's look at how this actually kind of works and how it's built up. They have a really nice little figure that kind of explains all the processes that go into this. So when you actually start constructing this, what you do is you kind of follow this path, and it will lead you to some sort of output that gets put onto the pretty GUI that you're interacting with. But there's a lot that's happening below. So you get some sequences that are all fed in, and you filter out some, you add some. What's really humorous is this include file actually includes the tiger from the New York zoo. I don't know if you heard about that, but this include file is like, hey, let's bring that tiger in because that's fun. So it goes filter, so you go and they clean up the data. So for example, when you're cleaning up the data and you're working with that, this is going to be a script they're going to put on the back end to do all that cleanup for them because they know what they want. So for example, they have too many ends, they want to clean up anything that doesn't look right. They'll play around with it, exclude some, and they filter out. This would be one of these scripts you'll see over here would be some of those that would clean up the stuff you don't want. So masking alignments would be how you get rid of some of the ends and stuff like that. So if you keep going, now we do an alignment. This is just making sure everything looks nice. But again, off to the side, they describe what's happening. If we look over here, this is going to be the actual scripts that are going to be written. There'll be something that'll be brought in, and there'll be some script that's written to take that big, huge chunk of data and cut it up into small parts because it makes the algorithm runs faster. Not going to get too much into that. We go through, we do masking. Again, that cleans up a bit. Okay, we go through, we simplify a little more. We figure out some stuff. What's really fun is they openly say that pretty much Oceania isn't very exciting. That's actually one of their comments because apparently not much has gone on to there. So again, another thing, getting context, doing subsamples. Because actually when you're seeing that big fun diagram, they can actually only display like 3,000 at a time, or it just gets too messy, so they drop some out, they add some more in, but they do in such a way that you're not really noticing. Okay, then a classic move happens in bioinformatics. Somebody built a tool to build that phylogenetic tree. So there's no need for me or these people to build a new algorithm. It already exists. So they will pull out something that exists, a property called IQ tree, feed it the data that they've produced above over here, and that will produce the tree. And they use another algorithm to prune it, which is just a way of taking off branches you might not want. And that's how the phylogenetics tree. This is a very common thing to do. There are properties that exist. Let's use those instead of making our own. Now, that's not saying that there's not bioinformaticians out there whose sole job it is to make these products. Like, I am going to build something that builds trees. That would also be a bioinformatician. But these are just people who are utilizing preconditioned trees. And then we keep following down the path. And we go and eventually get to a point where we have the output. We take whatever that output is, we spit it to the website, and it does all its pretty gooey magic. Okay, that I'm not going to get too much into because, yeah. So that's kind of the idea. And so if you follow down these, every time you see a little description, what's happening is they're either building a script to do it themselves, or there's a pre-existing tool that they're pulling from. And putting all of these things together, you build the back end that's doing all this analysis and doing everything, and then throwing it into something that looks really nice. So if we just look at, let's just pick a random code. So this would be the code that splits the sequence up into little pieces. So that would be right here. Okay, so step 2, alignment, it says break it up. So if you look through, this would be the code that would actually go and break up this code into smaller chunks. Now, you look at that, that seems like a really big task to do, but the code that does it is quite simple. It's, well, not simple, but it's condensed. And if you look at any of these scripts, so if you explore around, poke around, look at them, if you have a small understanding of computer science, you are probably able to look at this and kind of figure them out. You're just putting these tools together. You're building a puzzle, and that's what the computer science part of bioinformatics is. It's, hey, let's take these tools and kind of build a puzzle, and let's get this all working up in the background. So all these scripts don't look much more than this. They do small jobs, they get something done. And also a lot of these are in Python because Python is the language of choice for bioinformatics. So that's just kind of the quick dive into the code. I'm not going to go into anything specific because that's just a lot to dial down, and I don't know people's level of understanding. But this diagram, I think, shows that these are the stages. Every time you go off here, it's a description of what's happening. That's either a pre-existing tool or something they wrote themselves. You put the puzzle pieces together and you get, as Jessica phrased, the magic that's happening over here. Yeah, so that's kind of what I want to show you, and it's collaborative. So all of this you can see yourself, you can download. All of this stuff is information that you have access to, because again, a lot of stuff in bioinformatics is very open. Data is always being shared. It's really great that way. And that, I think, we'll end talking about that. So let's pick up the presentation for a big fun conclusion.
Ryan, I have a quick question for you.
Yes.
What's GitHub?
GitHub is just so it's a code repository. So it allows a lot of people in a lot of different places to all work on the same project. And what you do is there'll be code, you'll download it, work on it, upload it. So it allows multiple people to work on a project all at the same time from multiple locations.
And you can take it and use it in your own code encoding.
Depending what the copyrights are.
Okay.
So this one you're allowed to fully download, but depending copyrights may be different for different things. But yeah, so anybody can use GitHub as a project thing. So it's just a nice way to share information. Yeah, thank you. Okay, let's continue on. So lastly, let's talk about what we can grab from this. So if we saw the phylogenetic tree, we saw those branches, all those mutations, all those things are happening. And why is COVID-19 spreading? It's us. It's us traveling around the world. We take this virus that existed in one spot and we put it to someplace new. Then we brought it someplace else, and then we brought it to someplace else. And we can actually map how us traveling everywhere is actually the reason why, oh, my son just walked in, is why we actually have all of the coronavirus around the world. It's because we're traveling, and we actually have evidence that we can see that we are traveling because we have kind of the evidence in the code right here. It shows that we are traveling. It is spreading because of us. And excuse me for a second.
Yeah, no problem. I can take it from here. So the take-home message here is that although COVID-19 is showing mutations, the mutations aren't in any way, shape, or form a huge mutation rate. It's not leading to, as far as we know at this time, significant enough changes to the virus phenotype that we can call them different strains. We need to know more information before that can happen. But we can use these mutations to be able to make those lovely maps to get some epidemiological information and use that epidemiological information to make management and maybe even treatment decisions as we go forward. So with that, we wanted to open the floor to questions about the spread of COVID-19 and mutations as it stands. And you can use the chat function, as Ryan mentioned before, to ask your question, or you can turn on your mic and go ahead and ask the question aloud.
The first question was from Brian, asking, is that why they call it a data science pipeline? And yeah, you see very clearly that that's a pipeline. We just push the data in and information comes out the other side. And that's how a lot of this stuff is built, in such a way that you put all the moving parts together and you just start pushing data through.
Mm-hmm.
Yeah, that's one major aspect of bioinformatics.
And then the next question was, do you think there will eventually be different strains of COVID? Yeah, I do. Just like there is different strains of influenza, as the virus accumulates more and more and more mutations, there's bound to be mutations in regions that are coding regions, genetic coding regions, that lead to phenotypic changes. And those phenotypic changes might actually have physiological alterations that now you can call COVID-19 having two or more different strains to it. And Paul has said, yeah, I thought this is more into data science.
Yeah, it is, and bioinformatics in a lot of ways is a data science. You'll take the information that's being produced by a natural science like biology, and we'll apply computer science to do the analysis and all the work in the background. And that's data science and how it works. Yeah, so it's just a form, but it's just tied to biology specifically.
Mm-hmm. Any other questions about COVID-19? Or comments or something you might want to share if you've got some more knowledge than we do? Feel free.
Can you hear me?
I can.
Oh, I was just wondering, is open kind of like science, is this like a common kind of platform that's used in bioinformatics? Or are there many like platforms where people share data and?
Yeah, so Oh, do you want to go? Science is mostly a very collaborative field. A lot of people will publish, you'll do your data out, because all science is you do something and somebody has to be able to replicate it. So you have all of that happening. But as well, so a lot of this stuff's being shared, but we are in bioinformatics and that touches on pharmacy sometimes, and there's some industries that are a little less into sharing data because patents, stuff like that will also work into this. But yeah, a lot of the science is open, there's a lot of sharing and collaborative, but there's also some other stuff in the background as well.
Yeah, it's actually quite amazing how collaborative and open bioinformatics actually is. So this is just one example. NextStream is one example of a data repository along with bioinformatics tools that you can use and to visualize data. But there's ENCODE, there's the NCBI, there's
GenBank.
GenBank and OMIM, which is, if you want to know anything about any genetic disease that exists that we know about at this stage, and all of the literature around that, OMIM has that, and it's open access. So bioinformatics really is one of these open types of disciplines, multidisciplinary disciplines, that I think is really valuable in science. Mm-hmm. And part of the reason for why that culture has permeated is because initially sequencing was really expensive and only certain labs were able to do it. But the labs that were able to do to pay for the sequencing, do the sequencing, may not have had the same capacity to analyze the data or develop platforms. So there was this need for collaboration to occur. And so a lot of that early sequencing data, the Human Genome Project, for instance, was put out there in the world so that people around the world could figure out the most effective and efficient ways, and we can really use the resources in optimized ways. So, yeah. Any other questions before we move on to maybe the second part of why we're here today? Well, feel free to bring up other questions as we move along, but maybe we can move on to talking a little bit about our bioinformatics program. What do you think, Ryan?
I think that sounds like a great idea, and I'll stop sharing so you can start.
Oh, okay.
Because you're kicking this part off, and I'll grab the screen after that.
Sure. See if I can find it. There we go. And we're back. So, as many of you already know, Langara is very excited to be offering a brand new program in the fall of 2020. We're having our very first intake in the fall of 2020, and it's a baccalaureate degree in bioinformatics. So we do offer 2-year programs, but this is the first time that we're offering a 4-year undergraduate degree in science at Langara, and that's a very great opportunity for us, and I think a great opportunity for our students as well. So we start, I think we've got a good concept of what bioinformatics is, but I just wanted to bring this up once more, that when we look at the discipline of bioinformatics, it is the quintessential multidisciplinary field because it marries biology and computer science and mathematics and statistics in order to solve very complex problems in life sciences that we wouldn't necessarily be able to as just a human looking at data. So when Ryan pulled up that image, I'm so glad you did that, of the sequence of a viral genome, and it was just letters and letters and letters and letters. If I had to sit there and go through each one of those letters and compare it to another viral genome, I would make myself mad. It's impossible when we're thinking about comparing hundreds of genomes or one human to another human, where we have 3 billion base pairs in our genome, to be able to do that without the assistance of computers and mathematics and statistics. So bioinformatics helps us look at that data and parse the data, analyze it, make some sense to it, so that we can examine how our genes work, how they might fail, develop new strategies for diagnosis, develop new strategies for treatments to diseases. We can look at various forms of organisms that live in different environments to understand how they've evolved to live there and how they might be managed or manipulated to provide some kind of a function. For instance, if we look at mining industry uses a lot of bioinformatics right now, sequencing bacteria to determine if we can use bacterial functions to be able to clean up after tailings, for instance. So there's lots of opportunity within bioinformatics to be applied to various disciplines around the world. So why would you want to study bioinformatics? Well, I want to put it right out there. It's one of the highest paid science jobs you can get in terms of starting out with a bachelor's degree. One of the reasons is because everybody is looking for a bioinformatician right now. So these stats are a little bit old that we're looking at, but it's been projected that by 2020, the market for bioinformatics is growing and will reach over $13 billion. And much of that money is supporting people and people's salaries. When we look at the starting salary range for a bioinformatician that's just come out with an undergraduate degree, it's estimated between $47,000 to $53,000 per year. You don't see that kind of a salary range if you're graduating with a biology degree or a chemistry degree at that undergraduate level. So there's certainly money in the market, and there's money for people who are looking for entry-level positions. And there's lots of jobs. New jobs are being created in bioinformatics on a regular basis. And in fact, when the COVID scenario hit, just looking up online bioinformatics jobs, we saw lots and lots of jobs at that undergraduate level. People were really looking for bioinformaticians to help support virus research really early on in this pandemic. And then why would you want to take bioinformatics as a baccalaureate degree at Langara? Well, we worked really hard. Myself and a team of people, including Ryan, we worked really, really hard to create a curriculum that is applied, that's innovative, and that's collaborative. And it's a very unique program. You won't see a program like this offered at UBC or SFU. And in fact, we worked with UBC and SFU to create the curriculum for this program with their approval. So it's a very unique program. It's a collaborative effort. We also worked with industry partners to ensure that the curriculum matches what the demand will be with respect to employment later on. And I think I'm going to pause there, and I'm going to pass it over to Ryan to talk about the actual curriculum and what you might be studying. Ryan, you're muted.
I'm also trying to steal the screen again, which I think I did. I think this is mine now.
Success.
Hopefully. Yeah. Okay. So let's look at what this kind of looks like. So we're going to have two intakes, so a first and a third year. And depending what your experience is, you may fall into one of those two camps. But let's just look at what's happening as a whole. So this, as Jessica said, this is a very applied program. Here's why. All the courses you see, pretty much, they all have labs. So you're going to get a lot of hands-on experience. Not only are we going to teach you this stuff, you're going to actually work with it and play with this in the lab situation. And Jessica also said that this is a collaboration. So if we look at all the purple, those are all going to be our computer science courses. If we see the green, those are all going to be biology. Blue is going to be your chemistry. And all the, I'm going to say orange, is going to be your math and stats. So if you see, this is pulling from a whole lot of disciplines to teach you how to be a bioinformatician. And you need all this information to do that. And it's really, I'm excited for this program for one big reason is you're going to graduate knowing how to be a bioinformatician. Now, for me, I am a computer scientist. I have my degrees in computer science. I just so happen to really end up liking bioinformatics. So I had to teach myself all this biology after the fact. But you're going to come out not only appreciating comp sci, but also appreciating the biology that goes into it. So when you see a string of text that's a sequence, it's not going to be like, hey, look, an alphabet of characters. That's fun. You're like, hey, this is meaningful, and you have this good interaction understanding of it, which will lead to probably much better and more interesting actual analysis being done because you'll have a good understanding of both sides. That's enough about my love of this discipline. So as you see, this is how it would go. So you have your chemistry, biology, computer science, math, all that comes up. And right here, if you see, you're also going to walk away with 8 months of a co-op term. So this means you'll actually go out there, work in the industry, and get some experience. And that's very important to understand what's happening and being able to do that. And also, what's really fun and exciting is there's going to be a capstone project as well. And this means this is actually going to be a project you're going to do. So this is going to be something you may take off your work term. You may have a project you want to continue working on that may tie into there, or some other projects. This will allow you to get some research experience under your belt kind of in that way, or continue working on something a little more applicable that came from your job. Yeah, so that's the program in a nutshell, and yeah, I'm excited. Okay, so employment. So if you look at kind of the a word picture, I forget what these are called. Jessica, what is these things called?
Word cloud.
Word cloud. So if you look at the word cloud of kind of employment, the things you see are programmer, research technician, analysis. All of these jobs are what you're going to be doing. So a lot of the looking at data, getting it. Also, if you are and you end up liking to do a lot of the wet lab stuff, this is also opens up that as well. You would still be qualified to go out there, collect samples, run it through all of the sequencers, stuff like that is also something you're going to walk away with knowledge with as well. Okay. Okay, so here is another thing that kind of shows you that all because you decide to go down this path doesn't mean you have to go all the way. Now, hopefully, if you enter the program, you'll complete it, walk away, and be able to take this and go get a job in the field, or even take this into graduate study if you really want to continue on that way. Okay, but say you end up doing a couple years, it's not for you. Well, we also have that associate and diploma, and you can probably, after a couple years, apply for that as well and move on after 2 years if this isn't quite for you. And option 3, say you want to try this out, you take a few of these courses and you're like, this is not for me at all. That is fine. All the courses we're asking you to do are could be transferred to a university, which means you could take this and go into another associate degree or transfer directly to another university with the courses you're taking. Okay, so all because you come into this doesn't mean 4 years you're here, there's nothing, you're going to get nothing out if you don't finish the program. There's lots of these ways that if it doesn't quite go right, there's a lot of little exits you can do. Okay, so it's not, yeah. Okay, so that is the program in a nutshell, and this is when we'll take our second round of questions. So if anybody has any questions from the first part, or if anybody has programs about the degree in general, now would be the time.
You didn't even answer. I have a good one.
Go ahead.
Wait, I heard somebody.
Oh, I think your volume's not working very well. Maybe plug it into the chat. And while we're waiting for that person's question, I did want to point out that we have Ray Buenaventura here from the registrar's office. I'm calling you out, Ray, in case there's any questions about applications and admissions processes. So if you have a logistical question, we certainly have someone here that can help you with that.
I have a question in regards to the COVID-19 on how it's going to affect class sizes now. We briefly touched on this in the last information session. One moment, please, Richard. I'll be right with you, buddy.
That's my dad's kid.
Yes, it is.
Don't worry, it happens.
Yes.
I understand that most of the courses are now going to reduce class sizes as well as more of an online component next semester in the fall.
Mm-hmm.
Do we have a plan in place on how that's going to affect the syllabus?
There's a lot to unpack there. So I think I can say this because it's official that for the most part, the Langara is going to be going online remote teaching in the fall, which shouldn't impact the number of students that are registered in the program or in each of the courses. And anyone can jump in and let me know if I'm saying something that's not quite right, please. So the class sizes should be the same. Face-to-face components are we're still deciding what needs to be face-to-face, and the idea right now is that it's only going to be under very extreme circumstances that face-to-face will occur. So if there's a practical part or something of that sort. So for the program, I don't think it's going to make a big difference with respect to number of students per class, and for the most part, the courses are going to be remote teaching.
Thank you very much.
You're welcome, Matt. Any other questions? Oh, good, we've got some. Does that mean that we can start online and be physically present in Canada, but then join in person later? So depending on what happens with this pandemic, we'll have to revisit what's happening in the spring. But if we do start the program in the fall, you can be anywhere in Canada and realistically anywhere in the world and join online, start online, and then we'll revisit what happens with respect to face-to-face classes in the spring, and that information will be passed along to us by the provincial health authorities. It's not a Langara decision, it's going to come from outside of us. Good question, Kostya. And Paul, what a wonderful program. So unfortunate since I'm transferring to UBC this fall and they don't offer bioinformatics. Well, that's why you should come to Langara.
Yeah.
In this case, if I still want to continue to study at the graduate level, should I focus on studying biology plus comp sci, or to learn both math, comp sci, biology, and stat in a combination science major? Yeah, I think Ryan can probably speak to this because that's pretty much what you did, right?
Yeah.
Without a big bend towards biology.
Yeah, so you can always get into this going through computer science and focusing on that and taking biology in the background would get you, I would actually probably switch it to a comp sci major and a bio minor. But that's coming from me, and I'm a computer scientist. But I think depending how we talk to industry, that sometimes seems to be the more bend they like to see. Correct me if I'm wrong there.
Also related to your interests. Like if you are a biologist at heart like I am, my personal route would be to do all of the biology that I can and then add that sprinkling of comp sci so that I know how to program and I can use the tools. But if you are a computer scientist at heart and you want to build the tools and be at the cutting edge of that, then your focus should be on computing science and math with a flavoring of biology. I hope that helps.
So when it comes down to either choice you go with, you'll be okay as long as you have a good understanding of both of those topics and how they work.
Follow your heart when making that choice. And come to Langara. Any other questions about the program or about COVID-19? Oh, not to worry, Dennis, it happens to the best of us. Mics stop working in the middle of classes and it's par for the course these days, not to worry.
If I could just quickly add to Kostya's question about starting online. So there is a recent ruling by the Canadian government that you can essentially complete up to 50% of your degree online because of the COVID situation, and then still be able to qualify for your work permit, counting all those online courses. Because typically there is a limit in terms of the number of online courses you can use towards meeting your post-grad work permit. But in this current scenario, so we're hoping obviously that international students can come to Canada much sooner. Hopefully in the spring of 2021. But rest assured that even if you do decide to start online and start your program, in the fall term, If you are able to come into spring term, great. But if the situation does persist, you can continue to study online as well. So in terms of options, I think if you are considering coming to Langara, you can still definitely do that and have some sort of fallback plan with continuing to study online if you do have delays in your visa application, that kind of thing, beyond the fall term.
Yeah. Okay, one more thing to add to Paul's question. I suggest for comp sci, take enough that you can get to a machine learning course. If you can get up to there, that's probably enough comp sci, as Brian nods along. Yeah, if you can get to a machine learning course, you have known enough probably comp sci to get through most of bioinformatics.
And that can be applied to so many different disciplines. So the world would be your oyster if you can get there.
Yeah, it's an upper level course. It's typically, you're going to see that in a third or fourth year of most programs.
Any other questions?
Paul, I wouldn't say that one way is easier than the other way. They're both different. They're both different. Yeah. I wouldn't say one's easy, one's hard. I would say the transition is pretty good.
And it also depends. If you're more interested in the biology side of things, that would make it easier. And if you're more interested in the comp sci side of things, that would make that part easier. So really, it's a personal choice what you want to focus on. But yeah. Zilla, did you have a question? Maybe.
One last question. You mentioned in the last information session there was about 24 spots. Richard, your timing is impeccable. You mentioned there was about 24 spots. Now, is that annually, or is that only for the first intake? Do you have any expansion plans already in place?
So far, it's going to be 24 for first year every year that we offer the program, and that's yearly from our projections right now. And then the only time we will be doing a 24 student intake for third year is going to be the fall of 2020. That's not likely to happen again.
Yeah.
Okay, perfect. Thank you.
Oh, hi there. Can you hear me?
Yes.
Yes.
Oh, hi. I was wondering, sorry about earlier. There was just a lot happening in my apartment and my microphone was not off. But I was wondering, what would like a bioinformatics specific lab look like, as opposed to like a biology wet lab?
We've been to a couple. And I mean, I also have a job at the BC Cancer Center, and our lab is mostly wet lab stuff. So we've got wet lab docks, if you will, and benches. And then there's devoted areas for in silico or computer work. And I think a lot of bioinformatics that happens in those kind of hybrid types of labs. But then there's places like the Advanced Genome Center at the Vancouver Prostate Center, and there's the Genome Science Center, which are buildings full of computers. So it really depends on where you are. Good question, though. Ray, I was wondering if you can, if you have the information, if you can tell us about what's happening in terms of application deadlines.
Yeah, I got unmuted there. Yeah, so I think right now we are looking to extend the application deadline. So if people are interested in applying, the application deadline is set for May 31st. But I believe Jessica has extended that. So the application process is quite simple. I think Brian's already kind of gone through that a little bit, but it is on the Education Plan of BC website, so you apply online. In terms of what you would need, if most of you are already high school graduates, like you're not graduating this year but have already graduated, you can essentially put in your application and then submit your high school transcripts and other college transcripts, university transcripts if you have them, through the EPBC, the Education Plan of BC website, all in one go, basically. And actually, it's going to be a pretty quick process for us to actually complete your application if you have everything ready to go, and then send it off to Jessica, Ryan, and Brian for their assessment of your application. So it is a limited entry program, so it is competitive in terms of the application. I would say right now, you have a say pretty good chance of getting evaluated. So because you're not quite at the deadline yet. But it definitely is good to get your application as soon as possible, because we are, we do expect that there will be more applicants than seats. It is the first time around for a program, so one of the good things is that if you are interested right now, most likely the competition rate, so to speak, is not going to be as high as when we offer it the second or third time, when it really gets out there that there is a new program that offers this kind of high quality, relevant, for the job market, job ready kind of skills and training education that Bioinformatics does offer. So definitely, if you have any questions about the application process or about your application in general, you can certainly email admissions@langara.ca. And one of our advisors can get back to you. We're pretty much all online all the time, so we're pretty quick with getting back to you any questions that you may have. But yeah, feel free to do that. And of course, if you want to also, my email is pretty much, I think, on my screen here. So it's arbonaventura@langara.ca. So you can also just send me a note, and we'll help you out as best we can. So.
And while we're on the subject of emails, why don't I pop the Bioinformatics email in the chat. So if you do have questions and you're too shy to ask them right now, or that you've remembered it later, I'll give you an email address that you can send your questions to. Just want to make sure I typed it right. There we go. What else? Did we miss anything, Ryan?
I think that's everything.
We're super excited about this program. I know I am. If I had to go do it all over again, I say this all the time, I would take this program. I might be biased, but all right. Well, any last comments or questions? Well, we'll be holding another one of these virtual sessions on June the 2nd, same time, 4:30, same Zoom address. The topic will be something different. Well, still related to COVID and bioinformatics, but a different topic. And we will converge again and answer questions if you have them, and hope to see you in the program.
Yeah. Look forward to seeing you.
All right. Thanks, everyone, for the session.
Thanks, everyone.
Okay, bye.
Bye. We should probably stop recording now.
All right.